And then there's a bit of a transition where instead of talking about cloning DNA, instead you start talking about really more molecular biology techniques that are closer to those of biochemistry. And talked about is the use of gels. So gels are just what they sound like. If you think of Jell-O, Jell-O is a gel. The gels that we use in the molecular microbiology lab are not made of collagen, but they are the substances that are kind of squishy and fluid-like, but nevertheless have this structure to them. And what these gels have is a matrix of fibers all through them. And what this matrix is capable of doing is providing essentially a size filtration process or capacity. And this allows us to separate, if we can pull something through the gel, things according to their size. We can also, because the pulling process involves electricity, we can also differentiate organisms in terms of their electrical, not organisms, the things that we're pulling through the gel in terms of their electrical charge. But we can neutralize the charge basically by taking lots of stuff and mixing it with usually detergent, mixing it with, for example, a protein. And that way the properties of the protein other than its size are not really affecting how fast the protein moves through a gel. It's simply a mechanism of size separation. We can do the same thing with DNA. In fact, originally DNA sequencing was done on gels where we use size separation to determine the position of different lengths of fragments, and from that we can infer the sequence of the DNA that we're analyzing. But these gels allow for something known as gel electrophoresis. Polyacrylamide is the substance that's used to make the gels. It's a way of separating pieces of DNA from each other. It's a way of separating proteins from each other. It's a way of separating RNA from each other. And these separations are done for various reasons. One very common reason that it is done is so that we can determine whether a piece of DNA of a certain length or a protein of a certain size is present within some kind of mixture. So we can do some kind of an experiment and we can then see whether or not, for example, a certain kind of protein is present at the end of the experiment in higher or lower concentrations. Or when we're cloning, we can see whether or not the cloning vector, in fact, with the DNA inserted into it that we want to be cloning, is present within the organism that we're cloning into. And we would use a DNA gel looking for the plasmid in order to see whether or not, in fact, that DNA is present. If we're doing polymerase chain reaction, in order to detect the presence of a certain kind of DNA, we would do the PCR reaction, and then to confirm that, in fact, the new amount of DNA that we have is the specific DNA that we're interested in, we can then run a gel and see whether or not we have a DNA fragment that is of the size that it should be based upon the PCR reaction that we're intending to run. So gels are really important. Also described here as blotting. Blotting is a way of taking materials out of the gels, because the gels themselves are relatively difficult to work with, and put that material onto a substance that's easy to work with. The substance that's easy to work with is known as nitrocellulose, basically a filter paper type substance. And what you do in blotting is you in some manner pull the substance that is in the gel out of the gel and onto this nitrocellulose. The nitrocellulose is sticky to these molecules, so when you pull the stuff out of the gel onto nitrocellulose, it sticks to the nitrocellulose, and then once it's on the nitrocellulose, then you can do various kinds of analyses of what was on the gel. And what's nice about this is that not only do you have something that's easier to work with, but the position on the gel is retained on the nitrocellulose. Because you're actually taking this gel, this flat thing, and putting it onto this other flat thing and pulling the substances in the gel out of the gel onto the nitrocellulose, and so their positions on the nitrocellulose basically map to their positions in the gel. So we can determine differences in sizes as are indicated on the gel instead on the nitrocellulose. Now you don't always have to do this. It's possible to stain gels in such a way that you can see the different bands, as they're called, in their relative positions. But you can do more powerful things if you can pull the material off of the gel onto the nitrocellulose. What can you do on that nitrocellulose? Well, you can probe it. And probing involves taking certain kinds of molecules that are able to adhere to the substance that is found in the gels, and specifically adhere in a way that is highly specific to certain kinds of molecules. So for example, we can use DNA to probe, and that DNA that we're using will be complementary to DNA that's found in association with the gel, but not necessarily all the DNA in the gel. And in that way, we can see the location of specific sequences of DNA that we're using as our probe. With RNA, we can also probe with DNA, for example, and get a sense of where specific RNAs are located and in what quantities. If we were studying the RNAs that are produced by an organism, they would be mRNAs, messenger RNAs that are produced, and you can distinguish them. Interestingly, today we don't do that. We do much more often something known as RNA-seq. Basically, we just sequence all of the RNA that's present. We convert it into DNA, cDNA as it's called, and then simply sequence the DNA. And that gives us a much more complete indication of what RNAs are present, therefore what genes are being transcribed in an organism. With the DNA probes, the probing the RNA is called northerns. Probing DNA is called southerns. With the DNA probes, we can see whether or not specific pieces of DNA that we're interested in are present. So, for example, if we're trying to clone a gene, whether or not that gene that we're trying to clone really is in the DNA that we've isolated from an organism. And then there's something called westerns, and westerns, which you run on the gel, are not nucleic acids, but instead proteins, and then you pull those proteins onto this nitrocellulose, and you probe with antibodies, and the antibodies bind specifically to certain proteins, and therefore you can see whether or not your protein of interest, in fact, is present on the gel, and also infer how much protein in fact is present. So gels are an important component, less so these days than they used to be, but nevertheless an important component of analyzing organisms molecularly. They're also important for generating DNA fingerprinting, or fingerprints, which is a way of distinguishing among organisms in terms of their DNA. In this case, what we do is use these molecules that cut DNA called restriction endonucleases, and organisms that aren't clonally related are going to have specific sites that are cut by these enzymes in different locations, and therefore when you cut the DNA with these enzymes, you're going to get fragments that are of different size. And then you run out these fragments out of the gel and you separate them by size, and you can see differences in the array of fragment lengths from one organism versus another organism, and you can actually distinguish between individuals that are not clonally related. So in other words, you wouldn't be able to distinguish between identical twins, but you would be able to distinguish between siblings and in their DNA compared to someone that's completely different from them. You'd be able to distinguish even better because they'd have fewer cut sites in common. That's a restriction fragment length polymorphism. And you simply cut the DNA and then you run it on a gel, put it onto nitrocellulose paper, blot it onto nitrocellulose paper, and then probe it. And you'll get this array of bands, and the position of the bands will differ between different individuals, and they're clonally related to one another. Having said that, because DNA sequencing has become so expensive, these days it can be easier just to sequence an organism. organism. But sequencing takes more time than doing DNA fingerprinting. So if time is an issue, then DNA fingerprinting may be something that's worth doing instead. And let's see, what else do we have in here? Really, that's pretty much it. I mean, there's talk about various approaches to cloning, and there's talk about various products from cloning. But again, you can see there's a large cloning emphasis. But to repeat myself, these days molecular microbiology has a huge component of simply DNA sequencing. And in fact, these days, in terms of diagnosis, DNA sequencing and DNA sequences are becoming more and more important. And this is something that, because of when your textbook was published, is simply not an emphasis of the textbook. I'm sure if we looked at a newer version of the textbook, they'd be talking about things like metagenomics and the use of DNA technologies to identify different organisms or distinguish the different organisms and establish, for example, clonality. So that's all I really have, I think, that I want to talk about in terms of today's lecture. And it is still very much snowing out there. If anything, it's snowing harder now than it was when I began the lecture. Okay, see you later.